This is the Component 2 Taskbook Help Me video, and we are into applications of genetics. We've got the bacterium E. coli, produces an enzyme that can break DNA. The break always occurs where guanine is next to adenine. So where guanine is next to adenine. So that's where a break might occur. Write the bases. Okay, so you're just following your normal rules for pairing up the bases. Pairing up the bases there. Draw arrows to show where the enzyme would break the strands on 1 and 2, and then you're using this where guanine is adjacent to adenine. So again, the break would occur up here, but then you need to think about where is it going to occur on the lower strand there. And then just show us that separated strand after it's been cut. So leave the bits on the outside, just give us this middle bit. What's the most obvious feature of the sequence of bases in your drawing? Well, it means the drawing that you've just done up here or down here. You know, why would that base there link with that base down there? You know, what's the kind of rules that you follow in there? The region X, well, this is something that you definitely recognise the name. This is going to be the end, and there's a couple of different types of ends that you might get, but this is one type of them. And the group of enzymes, well, if you think about all the enzymes that are involved with your applications, any enzyme that can do things with DNA, it's one of those, and it's 2 parts, and 2 words to it. Part F, using circular DNA in a bacterium, and putting human DNA in there. So this is, if we have a bacterium there with a cell wall, you might have a larger piece of DNA, a single kind of chromosome there, but also there's small circles of DNA, small loops of DNA, and that's what we're talking about there. Name the enzyme used to fix the fragment into the bacterial structure. So this is think about fixing the fragment into the structure. So it's kind of joining up 2 parts, so that's the key, is joining up. That's the word that you should be concentrating on. And there's an alternative process where we're making the messenger RNA. So we've got some single-stranded RNA and then making DNA from it. So it's kind of like transcribing it, but the wrong way around. That's the clue there. And then turning a single strand of DNA into a double strand of DNA. Well, it's the enzyme that makes DNA, so it makes that DNA polymer. So that there's a clear word for that there. So pretty tricky. You're going to know all of those names of those enzymes and what they do, and that will help you with that. Advantage that this technique has, well, where are they collecting this from? Cells, but cells that produce that protein. So that cell is busy producing that protein, so is producing that messenger RNA, and not another one that does another job. So it's more specific but you need to sort of say specific for what, and just be a bit clear there. And finally, name the major international research project completed in the last few years, helping genetic engineers in the future. It's not the one with a number, but the one completed, just about the year 2000, that sort of time, and explain how it will help. That's 13 marks.